Okay, hello guys. So hope you are all able to hear me. So today we will continue the lecture of carbs. So we already explained the structure. We started with the metabolism. I will do kind of a brainstorming concerning the metabolism. So last time we mentioned that the glucose is the preferred source of energy for most of the body tissues. So brain cells derive energy from glucose. So the normal fasting plasma glucose is 70 to 100. Okay, guys, please try not to interfere. Try not to talk. عم شوف محمد وسالي ايه okay great محمد باتشي محمد حمزة okay great. Okay, so the normal fasting plasma glucose is 70 to 100. Okay, so that's why we need to avoid hyperglycemia, and we need to avoid as well hypoglycemia. So as we mentioned last time that hyperglycemia is risky, but hypoglycemia is life-threatening, especially that the brain cells derive energy from glucose. So last time we talked about glycolysis, and we mentioned that glycolysis is under aerobic conditions. So during glycolysis, if you remember, the glucose will pass through a series of steps to give by the end pyruvate. So glucose will consume 2 ATP to give me glyceraldehyde and 3-phosphate plus dihydroxyacetone phosphate. Here we have an isomerase. So by the end, in the next slide, you have to add 2 next to every single okay product. So here we have 2 NAD plus, 2 NADH, 2,1,3 biphosphate glycerate, 2 ADP, 2 ATP, 2 etc. So as a summary, so we will be having from 1 glucose, 2 pyruvate. We will be, as a summary, releasing or producing 4 ATP and expending or consuming 2 ATP with a total net of ATP 2, لأنه 4 minus 2 is equal to. So glycolysis is a total pathway where glucose plus 2 NAD plus 2 ADP plus 2 PI will give me 2 pyruvate plus 2 NADH plus 2 ATP. And as you mentioned, then glucose will give me pyruvate. This is the glycolysis. This is the first part of the metabolism. The pyruvate release will go and continue via Krebs cycle, or what we call citric acid cycle, to give me by the end CO2 and more ATP. So this step, we will see it later on. However, what we talked about is aerobic conditions. What about anaerobic conditions? We mentioned also last time that the pyruvate, either it goes to Krebs cycle or it can go and give me lactate, okay? Lactate under anaerobic conditions, okay? So, and this slide also was also explained. So last time also we mentioned that glucose will give me pyruvate. So what if I want to go just the opposite pathway? I want to go from pyruvate to glucose. What do we call this process? We call it the gluconeogenesis. Is it possible? It is possible. However, we will see that we do have some steps that are reversible, but we do have some steps that are irreversible. So basically, the irreversible steps are steps number 1, number 3, and number 9. And here comes the importance of having what we call key enzymes. So a key enzyme is an enzyme that will work in parallel, okay, to to overcome the problem of having an irreversible reaction. So in the step 1, instead of having hexokinase, we will be having glucose 6-phosphatase. In the step 3, instead of having phosphofructokinase, we will be having fructose 1,6 biphosphatase. And the step 9, instead of having pyruvate kinase, we will be having 2 key enzymes, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Okay, so here, if we just look at this figure, so from glucose to pyruvate, we call it glycolysis. From pyruvate to glucose, we call it gluconeogenesis. So let's look together and pinpoint where are the reversible and where are the irreversible. So step number 1, okay? Step number 1, from glucose to glucose 6-phosphatase. Is it reversible? No, it's irreversible. So this is the step 1. And here, instead of having hexokinase, we'll be having the glucose 6-phosphatase. This is the first key enzyme. What about step number 2? So here, it's reversible. We don't have any kind of problem. The same enzyme. So when I say reversible, that means that the same enzyme can be used both ways. Okay? So for the, as you can see, for the red arrow and for the blue arrow. So for the step number 3, to pass from fructose 6-phosphate to fructose 1,6-biphosphate, so here again, it's irreversible. So this enzyme will be replaced by another key enzyme. What about step number 4? Reversible, 5 reversible, 6 reversible, 7 reversible, 8 reversible. Here again, it's irreversible. So instead of having pyruvate kinase, here comes 2 key enzymes. So here we have 2 key enzymes to replace 1 enzyme. So instead of having this enzyme, we'll be having 2 enzymes. So through the usage of these key enzymes that we were talking about, 2 to replace for the step number 9, and 1 for 3 and 1 for 1. So here we can go from pyruvate to glucose, and this process is called the gluconeogenesis. Okay? Is it clear? Type please if it's clear. Lawan, where is the problem? Okay, perfect.
So, Doctora, can you just tell us again about the irreversible and the reversible? Like, what's the difference between the reversible and the irreversible?
Okay, so here, as you can see, for example, for this arrow, when you have a double arrow, اوكي شوف هون كيف عندك double arrow, يعني that's mean انه انا فيي اروح through using this enzyme I can go from here to here or I can go from here to here. So I don't need another enzyme. So here you can see that we are using the same enzyme to go from 1 till 2 or to go from 2 till 1. So this is called reversible. Okay. However, from glucose to glucose 6-phosphate, it's a one-way reaction. I need hexokinase. So if I want to go from glucose 6-phosphate to glucose, I cannot use hexokinase because this reaction is not reversible, it's irreversible. So here I need another enzyme allowing me to go from glucose 6-phosphate to glucose. Is it clear, Lawan?
Yes, it's clear.
It's the same for step number 3 and for the step number 9, okay? So, and here we should know that mainly the gluconeogenesis will occur in the liver, okay? Because these key enzymes are present there, okay? What about the ATP? So here we have 3 enzymes, the pyruvate carboxylase, the phosphoenol pyruvate carboxykinase, and the phosphoenol glycerate kinase, 1 ATP each. So 3 ATPs are used by 1 pyruvate to produce 1/2 glucose. So let's look at where is the enzyme. So pyruvate carboxylase. Where is pyruvate carboxylase? It's here. So pyruvate carboxylase, okay. Pyruvate carboxylase require 1 ATP, okay. And phosphoenol pyruvate carboxykinase, this one require 1 ATP. And phosphoenol glycerate kinase, where is phosphoenol glycerate kinase? Here, okay. So phosphoenol, these 3 enzymes, كل وحدة منهم بدها 1 ATP. So in total require 1 ATP. So 3 ATP will be used by 1 pyruvate to produce 1/2 molecule of glucose. Who can tell me why we are saying 3 ATP are used by 1 pyruvate to produce 1/2 molecule of glucose? Why not to produce 1 molecule of glucose? Why 1/2? Who can tell me? Hello, guys.
Just a moment.
Okay. What about the other ones? Why you are that passive? Maya, Lamis, Reem. Who can tell me why I'm saying that 1 pyruvate use 3 ATPs?
Because actually, if we look back at the to the pyruvate carbox, like the final one, it is it's actually half, like it's actually half of the reaction, okay, at the end of reaction.
So like, simply it's because 1 glucose will give me 2 pyruvate, right? Do you remember the glycolysis? Yes. 1 glucose gives me 2 pyruvate. So if I have 1 pyruvate, if I want to go back, 1 pyruvate will give me 1/2 glucose. So to produce 1 glucose, what do I need? I need 2 pyruvate, right? If I need 2 pyruvate, if every single pyruvate requires 3 ATP, so 2 pyruvates require 6 ATP, right? Is it clear? So again, I will explain.
Yeah, please.
Okay, so one glucose, okay, one glue will give me, okay, 2 pyruvate, right? 2 pyruvate, right? Okay, so one glucose will give me 2 pyruvate. If I want to go back, if I'm telling you that to pass from 1 pyruvate, okay, I have 3 enzymes in the gluconeogenesis. Every single enzyme requires 1 ATP. So in total, 1 pyruvate requires 3 ATP to give me half glucose. So to have 1 glucose, what do I need? I need 2 pyruvate, and automatically I need 6 ATP. Clear? It's not difficult. Clear? Yes? Okay, perfect. So I can...
Yes.
Okay, Lama?
Yeah,
So,
I understand it clearly.
okay, perfect. So what about the substrate for gluconeogenesis? What do I mean substrate? Substrate is a product that will launch a reaction, okay? So what are the substrate? One of the substrate is lactate. Why? Because we said last time that, we said in this lecture also, that glucose will give me pyruvate. Pyruvate can give me also lactate. So, and we said that whenever I have an excess of lactate, the lactate can give me back pyruvate, and automatically the pyruvate will give me back what? Glucose. So again, lactate will give me, glucose will give me pyruvate. So pyruvate, either it will go Krebs cycle or it will go lactate if it's anaerobic. So the excess of lactate can either go back and give me pyruvate, and this pyruvate can either go Krebs cycle or can go back to give me what? Glucose, okay? So here also we have alanine and glycerol. I won't take time now to explain alanine and glycerol because we will see later on how alanine is linked to gluconeogenesis, okay? Again, what are the hormones of gluconeogenesis? So who knows what is the role of insulin? Who has one of his relatives? So insulin, does it increase the blood glucose level or does it decrease the blood glucose level?
So the insulin will take the...
So the insulin will increase the blood glucose level. The insulin. I will type the question. The insulin. Okay, one moment. I will try to say it.
Right.
Oh. So the insulin will increase the blood
decrease أكيد.
level
أكيد
or
decrease. Inhibit it, like it will
decrease.
It will decrease.
It will decrease. It will inhibit. So if the insulin will decrease the blood glucose level, so do you think the insulin will enhance the gluconeogenesis or will inhibit the gluconeogenesis? Who can tell me?
Inhibit.
Inhibits the gluconeogenesis. Perfect. Why? Because in the gluconeogenesis, we are more transforming the pyruvate to glucose. So if I don't want to go this way, so the insulin will inhibit the process. The glucagon and the glucocorticoids are hormones that work just the opposite way of insulin. So in this way, glucagon and glucocorticoids will increase the gluconeogenesis to give me more what? More glucose or more pyruvate?
Can you repeat, please?
So we are saying that the insulin, more glucose, exactly, perfect. So the insulin will inhibit the process, but the glucagon, since the glucagon just works the opposite way of insulin, it will increase the gluconeogenesis to give me more glucose, okay?
Yep.
Great. So physiological significance. So the major metabolic significance of gluconeogenesis is to maintain the blood glucose level under conditions of starvation. So that's mean if I'm under starvation, if I'm fasting, okay, and just the opposite way, if I'm eating too much, in both ways, we should have a metabolic reaction maintaining the blood glucose level to its normal fasting rate, which is 70 to 100. Agree? So we are saying if I'm at the level of starvation, okay, we do have some processes that should be stimulated. One of these processes, the under starvation, automatically my blood glucose level will decrease or increase. If I'm under starvation,
Decrease.
it will decrease. So automatically gluconeogenesis should be stimulated or inhibited.
Simulated.
Stimulated, exactly. So here we are saying that under the conditions of starvation, the gluconeogenesis are stimulated. Thank you for the responses. Yeah, I can hear the responses. They are all correct. So the brain has a minimum obligatory requirement of 120 grams of glucose per day. So that means that I need to have a minimum of 120 gram of glucose per day to be able to maintain my life, okay? So under the condition of starvation, one of the processes that will help me to maintain my brain glucose, blood glucose level is what gluconeogenesis, okay? But we'll see that we do have other processes that I'm going to talk about also later on, okay? So one of these processes is glycogenolysis, okay? So again, glucose to pyruvate, glycolysis. Pyruvate to glucose, gluconeogenesis. But here comes the storage form of glucose. What do we call it? The animal storage form of glucose. It is glycogen. So glycogen is the animal storage form of glucose, okay? So if I need to increase my blood glucose level, what should happen? The glycogen should be broken down to give me more glucose, right?
Isn't this like the T1 and T3 when we have the high in metabolism, like thyroid?
Kind of. هلا بس
Oh,
don't talk about thyroid. Let's talk about glycogen. So glycogen is the animal storage form of glucose. So if I'm fasting or I'm starving, the glycogen is glucose, okay? So glucose linked together. So what should happen? If I'm starving, my blood glucose level will decrease. So
will
what
decrease.
will do the glycogen? The
S-
glycogen should be broken down. So this reaction is called glycogenolysis. So glycogenolysis, so lysis, glycogenolysis or glycogenolysis, lysis means destruction or breaking down. So here, when I need to lift up my blood glucose level, the glycogen will be broken down in order to raise the blood glucose level. And when I'm eating in excess, okay, so I have more blood glucose level, what should happen? I need to decrease my blood glucose level to reach the normal fasting state, which is 70 to 100. In this case, okay, the excess of a glucose will be conserved and stored under glycogen. And in this case, we call it glycogen synthesis, okay, or glycogenesis, okay? So I will explain again, okay, للي ما فهموا. لكن
please.
حكينا نحن we pass from glucose to pyruvate, glycolysis, from pyruvate to glucose, gluconeogenesis, okay? So now we have other processes that are being involved. If I'm eating, okay, too much sugar, if I'm eating too much sugar, okay? So at a certain point, I'm risking my blood glucose level to be more than 100. So what my body should do to maintain it 70 to 100, I need to figure out a way. The way is to store the excess of blood glucose level under glycogen. Okay? شو بسميها glycogenesis. So بيجوا هول glucose بيعملوا link together and they form glycogen. It's the storage form. And now I'm fasting. I'm fasting for a whole day. So I need to figure out a way to raise my blood glucose level. So the animal storage form of glucose, which is glycogen, will decide to be broken down under a process called glycogenolysis, okay, to give me what? To give me glucose to lift up my blood glucose level. Okay? Clear? Please type if it's clear.
Yes, it's very clear.
Okay, perfect. So here, glycogen is the storage form of carbohydrates in the human body. So the major sites of storage of glycogens are liver and muscle. Okay, so liver and muscle are the basic sites of glycogen storage. So the major function of liver glycogen is to provide glucose during early onset of starvation. Okay, so it's really important to know that during starvation, the liver glycogen. So the glycogen is mainly stored in liver and muscle, but the liver glycogen basically during starvation will be broken down to lift up my blood glucose level. So again, when blood glucose level lowers, liver glycogen is broken down and has to maintain blood glucose level. Okay, so after taking food, blood sugar tends to rise, which causes glycogen deposition in the liver. After 5 hours after taking food, again the blood sugar tends to fall. Then the glycogen will be lost to glucose to meet the energy required. Okay, so it's like a balance. Okay, if I have an excess of a glucose, no, it's alarming. I need 70 to 100. Go and store yourself under glycogen. And now I didn't eat like for 5 hours. So no, I need more glucose. Break yourself glycogen and give me back glucose. Okay? Questions or clear?
Yes. So does this occur when we are sleeping, for example? Because like
Yes.
when we are sleeping, it's 8 hours, and we
But
are
yeah,
not
but
eating.
when you're sleeping, you don't need that much energy. So definitely during the day, so you need more energy, so it will be more active. Okay?
Okay, thank you.
Okay, welcome. So here we will talk about glycogenolysis or glycogenolysis. So what do we have exactly? So this is the famous structure of glycogen, okay? So it's a glucose, a glucose, a glucose, a glucose, a glucose, a glucose. What will happen? So first of all, we have the glycogen phosphorylase, okay? So the enzyme glycogen phosphorylase removes glucose unit, okay? So as you can see here, so this glucose unit, okay, was here cut. And instead of having O, since it's phosphorylase, what it will add? A phosphate group, okay? So here you have to imagine that, okay? I will use the pen to show you. So here, tac, we cut here. So we cut here, so we debranch the glucose from the glycogen, okay? And we add a phosphate group. Why a phosphate group? Because this enzyme is called phosphorylase. We are adding a phosphate group. And here we will be having a glucose 1-phosphate. Why 1? Because it's at the position 1. And then we do have another enzyme called isomerase. This enzyme, okay, will pass the phosphate from position 1 to position tag 6. So here you can see that the phosphate was passed to here. And then we have another enzyme called the glucose 6-phosphatase. So when I say phosphatase, it means it will remove a phosphate. So H2O will take the P to give me another compound to take off the phosphate and to give me finally the glucose, the free glucose that I want, that will be released where in the blood to lift my blood glucose level. Okay, should we memorize this? What you should memorize is as follows. So we should know that in case, okay, the blood glucose level is under starving conditions or under fasting conditions, so we need to lift up the blood glucose level. Then the glycogen will perform glycogenolysis, where first of all, yes, you need to know the enzyme. The glycogen phosphorylase will cleave a bond to release a glucose with adding 1-phosphate. Another isomerase will pass the phosphate from position 1 to position 6, and by the end we have a glucose 6-phosphatase to be able to remove the phosphate and release a free glucose. Okay, so you can memorize it as I'm explaining it. You can take note later on. Okay. Yola, I already mentioned the summary, okay? So here you should know that the glucose 6-phosphatase is only present in the liver. So that means that the glycogenolysis can only happen in the liver, okay? So hepatic glucose 6-phosphatase will do this reaction in the liver, okay? So now let's pass to the factors, okay, regulating the blood glucose level. Okay, so here, as you can see, we have low blood glucose and we have high blood glucose. So let's start by the option low blood glucose. If we have low blood glucose, which hormone should be stimulated? Who can tell me? Yalla, guys.
Can you repeat the question, please?
So if we have low blood glucose, which hormone should be stimulated? Glucagon
alpha
or
cell,
insulin?
uh,
Glucagon or insulin?
glucagon.
Glucagon, perfect. Glucagon, okay. So, and about the process, which one should be stimulated? A glycogenesis or a glycogenolysis?
Glycogenesis.
If I have low blood glucose, I will synthesize the glycogen or I will do glycogenolysis?
Glycogenolysis.
Glycogenolysis, perfect, Sally. Glycogenolysis, okay.
I said
So
it too.
now, no, you said glycogenesis. So if we have high blood glucose, now which hormone will be stimulated more and released? Insulin or glucagon?
Insulin.
Great, insulin. And what about here the process is if I have high blood glucose, so I will synthesize more glycogen. It's called the glycogenesis. So now we will see more the factors that will enhance the entry of a glucose in our blood. Perfect, guys. Okay, all the answers are correct. So what are the factors enhancing the entry of glucose in my blood in order to increase my blood glucose level? First of all, absorption from intestine. So we should know that if I'm eating more, definitely I will increase my blood glucose level, right? So second, glycogenolysis. If I'm breaking down glycogen more, so I will increase my blood glucose level, right? Number three, gluconeogenesis. If I convert more pyruvate to glucose, then I'm increasing more my blood glucose level. And number four, hormones, hyperglycemic hormones such as glucagon and glucocorticoids. So these four factors mainly will cause the entry of a glucose and the increase of blood glucose level. So now what about the depletion of a glucose? How can I decrease my blood glucose level? Number one, if the tissues of my body are using the blood glucose, I will decrease the blood glucose. Number two, if I want to synthesize more glycogen, I will decrease the blood glucose, right? Number three, if I want to convert a part of the glucose into fat, this is called lipogenesis. So in this case, also I will decrease the blood glucose. And number four, if I'm releasing more insulin, I will decrease my blood glucose. Okay? So now we will see a scheme summarizing all what I talked before. Okay? So please concentrate with me, guys. Now we will see, okay, where we have two arrows, okay? We have one red and one blue, clear? So the red one is for activation and the blue one is for inhibition, okay? So we have A and we have B. So in the A, so these two schemes, first of all, they do represent the regulation of a blood glucose, okay? So A in the fasting state. In the fasting state, what will happen? So first of all, if I'm fasting, so as you can see here, the absorption is inhibited. Definitely, I'm fasting. So I'm not eating. So the absorption from the gut, from the intestine will be inhibited. Okay? So, however, here we can see that the glycogenolysis will be activated in red. I need to break down a glycogen. The gluconeogenesis will be also activated. I need to convert more the pyruvate to glucose to lift up my glucose level. Here, as we can see, the lipogenesis will be inhibited. Why? I don't have enough glucose to convert it to fat. I cannot do this. I'm already in the fasting state. So this will be inhibited. But the lipogenesis will be inhibited, but the lipolysis will be activated. So here, the fat will try to, so it's free fatty acid, FFA. So the fat will try to do what is called lipolysis. So when you see L, Y, SIS, so lysis, yani destruction. So here the lipids will tend to be destroyed into free fatty acid to give back energy as a secondary state. If you remember, I said before that carbs are the first energy provider, but lipids are the second energy providers. So here in this case, and that's why, as I told you before, so with starvation, we rely on carbs. We try to maintain, we try to do more glycogenolysis, to do more gluconeogenesis, to lift up the blood glucose level. But at a certain point, we are not able to manage, we are not able to handle. So what will happen in this case? We will be having more lipolysis. Why? Because the lipids will start to be broken down into fat to give us more energy. Okay, so this is what is called lipolysis. من هيك ببلش الواحد يضعف لأنه بصير الفات بده يدوب تيعطيه energy. Okay, so is A clear for you? Please.
Yes.
A. What about the others? Yeah, clear? Perfect. So what about B? So B, it's not fasting. So B, it's another scenario where the glucose level is very high. So I'm eating. So first of all, as you can see, the absorption will be activated. I'm eating, it's normal, but I'm eating in excess. So my blood glucose level will be very high. What should I do? Number one, glycogen synthesis. Go and store yourself under glycogen because I need to maintain 70 to 100. I don't need that much glucose. Number two, okay, lysis يعني destruction يعني breaking down. Okay, so number two, I should have more glycolysis. Okay, so here we can see that the gluconeogenesis will be inhibited. I don't want to convert more pyruvate to glucose. I inhibit. It's in blue. Okay, here the lipogenesis will be activated. Why? I have an excess of sugar. Let's go and store it under fat. Let's store more fat, so lipogenesis will be activated, but the lipolysis will be inhibited. I don't need to break down lipids to give me an alternative of energy. I have an excess of glucose, I have an excess of energy. So is B clear? Do you have any question? Okay.
So
Omar,
this is how a person can get fat.
Omar, where do you have a problem? Please, can you go online and speak to me? Tell me what is your problem? No problem. Okay, perfect. Okay. Lawan, do you have any question?
Yes. So in the P part, so you said we stored a lot of the fat, like when we have the right amount of the glucose. So then we're gonna change it to the.
When we have an excess of a glucose, so the glucose tends to react with free fatty acid to have a storage of fat. Okay?
Okay.
We call this lipogenesis, okay?
Okay. So, okay. Got it. Thank you.
Okay. So here also another scheme to summarize the main idea, okay? So here we have left side all the hyperglycemic factors and right side all the hypoglycemic factors. So what is hyperglycemic? شو يعني hyperglycemic? That will increase the blood glucose level. So gluconeogenesis will increase the blood glucose level. Glycogenolysis will increase the blood glucose level. Absorption from the GIT, from the gastrointestinal tract through eating, it will increase. And also hormones such as glucagon, corticosteroids will increase the blood glucose level. So what about the hypoglycemic factors? So glycolysis will decrease the blood glucose level. Glycogen synthesis will decrease the blood glucose level. Lipogenesis, so synthesis of fat and fat deposit, will decrease the blood glucose level. And finally, insulin will decrease the blood glucose level. Clear?
Yes, it is. It's much more clear now.
Perfect. Okay. So what about the normal plasma glucose level? So as I mentioned before, it should be 70 to 100. So fasting states means the glucose is estimated after an overnight fast of 12 hours. So when I say fasting means I fasted for 12 hours. So following a meal in a normal person, the glucose level does not usually rise above 140 due to the prompt secretion of insulin. So insulin, as we all know, will regulate directly the blood glucose level. So when the blood glucose level is normal, we call it normoglycemia. So when it's high, we call it hyperglycemia. And when it's low, we call it hypoglycemia. Okay? So when values are above the normal range, we call it hyperglycemia. When values falls below 50, we call it hypoglycemia. So it's a Greek term. Hyper means above and hypo means below. So as I mentioned before, also hyperglycemia is harmful, but hypo is fatal and life-threatening. So what about the sugar in urine? You should know that by default, normally glucose is not excreted in urine, okay? But when the person is diabetes, we have what is called a renal threshold. It's a value. It's a value that will, starting from this value, we can have a glucose in urine. So for the diabetic person, usually, whenever the blood glucose level will reach 180, so if the blood sugar level is more than 180, this is called renal threshold. يعني the value that will إذا بتكون يعني بتفرق بين to have or in the presence or absence of glucose in urine. So the renal threshold is 180. So above 180 milligram per deciliter glucose in our blood, we will be having glucose in urine. But for the normal person, we do not have glucose in the urine. Okay. So oral glucose tolerance test, it's a type of test that we do to check the level of glucose. So how do we do it? So usually the patient is instructed to have good carbs diet for 3 days prior to the test. So he should abide by a diet rich in carbohydrates. The patient should not take food after 8 p.m. Okay. ما بياكل بعد 8 وما ياخد breakfast. So بيكون عندنا 12-hour fasting. So هم بنكون أخذنا 3 days rich of carb, بعدين 12-hour fasting, and about 8 a.m. after the fasting, we take a blood sample, okay? And we also can have urine sample. And after the sampling collection, we analyze. Let's go directly to the result that is interesting. So here, just look at the table and look at the renal threshold. So the renal threshold, as you can see, it's 180. Okay, so for the normal person, it's below the threshold. So here, for the normal person, you can see the peak. Okay, for the diabetic person, just look, it is above the level. For the person that take a diet very high in the glucose, it can be also above. What you should memorize, you should know, it's not really important, this test. What we should memorize is that the renal threshold of having glucose in urine is 180. But here, just as info also, you can look normal person, criteria for diagnosing diabetes, okay? So we don't really care about the last column, it's too advanced. So here, as you can see, so for the fasting, normal, less than 110, okay? For the so for 1 hour peak after the glucose 160 for the normal, 2 hours after glucose 140. However, for the diabetic person بصير 200 يعني عنده كمان high blood glucose level وعنده high urine presence of glucose. Okay, so now I will pass to the chapter 7. So what's really important is mainly the schemes that summarize the hyper and the hypo. So we should know what are the hyperglycemic factor, what are the hypoglycemic factor, how can we control, okay, so when we are fasting or when we are taking an excess of sugar. So now we will pass to the minor metabolic pathway of carbs. So we should know that by default, the carb will abide by what we talked about, يعني glycolysis, gluconeogenesis, يعني Krebs cycle, etc. However, sometimes, okay, we can have a minor pathway. So it's like a deviation. What an all minor pathway, it's a deviation. So this pathway is called pentose phosphate pathway أو hexose monophosphate pathway. So instead of a glucose, بدل ما الجلوكوز يروح through glycolysis, بيقرر to shut down and to go through another pathway that we call pentose phosphate pathway. You should know that about 10% of glucose molecules per day can enter this pathway, and the liver and the red blood cells metabolize about 30% of glucose by this pathway. So the major purpose of this pathway is the generation of reduced NADPH and to also to release pentose phosphate. Why do we need pentose phosphate? To synthesize nucleotide. Do you remember nucleotide? Omar, what you did not understand? Or let me finish this. Yeah.
I think he didn't understand the OGTT.
Okay. So OGTT, it's an oral glucose tolerance test. Okay. So بتقول له للبيشنت ياكل 3 ايام اشياء فيها كثير سكر، بترجع بتقول له يعمل فاستنج، بترجع بتاخذ منه شو اسمه sample. Okay. blood sample. And here, as you can see, وبتاخذ منه urine sample. So here it's the urine sample. So نحن قلنا انه here you can see 180. 180 is the renal threshold. So look, for the normal person it was very low. لأن normal person ما عنده glucose بالurine. بس for the diabetic person كان high 250. يعني هو عنده glucose باليورين. ليه؟ لأنه he crossed the renal threshold اللي هو 180. Okay. Apparently he's not online anymore. Anyway, let's go back to the pathway. Okay, you can always send me questions later on. So about 10% of glucose molecules do not undergo glycolysis. They do undergo a pentose phosphate pathway. Okay, so why it is important to have the pentose phosphate pathway? Mainly to release what the pentose phosphate. Okay, the pentose phosphate that we are going to use as nucleotide. Okay, let's have a look directly at this scheme. So this is the glucose 6-phosphate. Okay, do you remember the glucose 6-phosphate? It's the second step after glucose. So in the glycolysis, we have a glucose and we have then a glucose 6-phosphate. So here, the glucose 6-phosphate, under the reaction of an enzyme, here you should add the glucose 6-phosphate dehydrogenase. Oh, let me write it like this. Here, we should have an enzyme called glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase. So the glucose 6-phosphate dehydrogenase is an enzyme very important in this pathway. Okay, so this enzyme will convert a glucose 6-phosphate to 6-phosphogluconate. Okay, and then through the assistance of NAD+, we will be releasing CO2 and we will be releasing ribulose 5-phosphate. So this is called oxidative phase, but somehow for X reason, the ribulose 5-phosphate decide to go back to glucose 6-phosphate. And if it will give بنسميها high non-oxidative. إذا أعطتنا glucose 6-phosphate, the glucose 6-phosphate is an intermediate in the what? In the gluconeogenesis. لأنه عندنا glucose 6-phosphate. So if I have more glucose 6-phosphate, what it will give me back? Glucose. So that's why here you can see that during the non-oxidative phase, the pentose phosphate is converted to an intermediate of glycolysis. So it will give me back a glucose 6-phosphate that will give me back a glucose. If not, in the oxidative phase, the ribulose 5-phosphate will give me ribose 5-phosphate, and the ribose 5-phosphate, I can use it to what? To form nucleotides, the base of DNA and RNA. And here we should know that in this reaction also, we have oxidized glutathione. This is called oxidized glutathione. This is oxidized glutathione. And here we have the reduced glutathione. Here we have the reduced glutathione. Okay, so oxidized glutathione will be reduced by NADPH to reduced glutathione. So this is very important because reduced glutathione are very important, okay, to combat the oxidative stress, which is very bad, the oxidative stress for us. Okay, لكن if you want, I can tell you what to write. Can you take notes?
Yes.
Guys, can you take notes? Okay, so let's start. Okay, so let's say that. Okay, ready?
Yes.
Okay, so you can say that usually the glucose undergo glycolysis, which is the regular pathway of carbohydrate metabolism. However, 10% of the glucose, 10% of the glucose undergo a minor metabolic pathway, called pentose phosphate pathway, Okay? So, in this pentose phosphate pathway, we do have two phases. In this pathway, we do have two phases, oxidative and non-oxidative. In the oxidative phase, so in this pathway, we do have two phases, oxidative and non-oxidative phase. In the oxidative phase, the glucose 6-phosphate, will be converted to 6-phosphogluconate under the activity of an enzyme, or catalyzed by an enzyme called glucose 6-phosphate dehydrogenase. Okay? So, under the activity of this enzyme, the oxidized glutathione, which one, Lawan, is the oxidized glutathione? Lawan?
GSSG.
Okay, so the oxidized glutathione, GSSG, is reduced, the oxidized glutathione, GSSG, is reduced by NADPH, to the reduced glutathione, GSH, to the reduced glutathione, GSH. The reduced glutathione is required to combat the stress, and maintain the normal reduced state in the cell. So this is the normal way. So the normal way, we should have more reduced and less oxidized, okay? So afterwards, the 6-phosphogluconate, will generate ribulose 5-phosphate, that will give the ribose 5-phosphate, important to form nucleotides. So the main objectives of the pentose phosphate pathway, is the generation of pentose phosphate for nucleotide synthesis. Is the generation of pentose phosphate for nucleotide synthesis. Okay. هلا بدنا نقول انه exceptionally the ribulose 5-phosphate can go into a non-oxidative phase, to give me back glucose 6-phosphate, which is an intermediate of a glycolysis that can give me by the end glucose. Okay. طيب guys, I will stop here. Okay, please بدي أقول لكم إنه attendance will be taken. Okay, so يلي كتير طلعوا online و offline. Okay, please on Monday try to be all the time online لأنه I will take attendance. Okay, بدي منكم to read to review هلا. It will be recorded the session. Okay, watch it one more time. Okay, Monday we will continue this lecture and we will start by the lipids. So I can kindly ask you please to read the remaining part of this lecture and to read the first part of lipids. Okay, so see you on Monday at 10. Feel free if you have questions to send me the question. Thank you for attending. I know it's very difficult. It's not that easy, but صدقوني, it's much more better than to skip more and more lectures. Then it will be very easy to make makeup sessions for all the missed parts. Okay, do you have any question? Thank you so much. Stay safe. Okay, thank you again.